Hello and welcome to my channel Omni Bioentrance, and today we will be continuing with the IIT JAM Biotechnology question paper conducted in the year 2025. So coming to the first question in this I will be discussing mainly the numerical aptitude questions, mainly the biology questions. Physics, chemistry, maths I am not discussing. So coming to the first question, free energy change for transport of an uncharged solute across a membrane against a uh, uh, 1 into 10 raised to 3 fold concentration gradient at 25 degrees Celsius is. So free energy change they have asked. We know the formula delta G is equal to RT ln C2 by C1. C2 is the concentration outside and C1 is the C2 is the high concentration. 1 minute. For the transport of an uncharged solute across a membrane against a 1 into 3 fold concentration. Okay. Yes. C2 is the concentration inside. It is inside, it is 1 into 10 raised to 3 fold. And C1 is outside. We will take it just like as 1 only. 10 raised to 3, it is 1000. I have written as 1000. Okay, so free energy for transporting an uncharged solute against a concentration gradient, it is given by delta G is equal to RT ln C2 by C1. We have to convert this 25 degrees to Kelvin. See here R value is in Kelvin. And here all okay, so here convert by adding 273.5, you get 298.15 Kelvin. Substitute in this value, natural log of C2 by C1 is 6.90775. Substitute the value for delta G, you will get it as joule per mole per Kelvin. Everything is in joules. That is especially the R value, T is in Kelvin. This is concentration. So you will get the answer in joule per mole. Convert, the answer is in kilojoule per mole. So you have to convert by dividing by 1000. You will get it as kilojoule per mole. Then next one, a forest has 4 different species and their numbers are given here. Shannon Biodiversity Index. So it is given by the formula H is equal to sigma S I is equal to 1 Pi ln Pi. Now S is the total number of species in the ecosystem. So add up all these 60, 20, 10 and 10. You will get the total number of species, that is 100. Then Pi, it is the proportion of the species in the community. That is in community A, in B, in C, in D. Proportion of species of A, B, C and D. Species A, B, C and D are species. I made a mistake by telling it is community. So proportion of species in A, B, C and D. So A species, it is 60 numbers. So you have to calculate Pi. Pi is equal to Ni by N total. N total you already know, that is 60 plus 20 plus 10 plus 10, that is 100. Ni is for A it is 60, for B it is 20, C it is 10 and D it is 10. Substitute the values and you will get Pi. Pi is equal to Ni by N total. You will get these values. Then after that you substitute in the formula. Don't forget the minus sign outside the bracket. Okay. So it is Pi ln Pi. So see 0.6 ln 0.6 plus 0.2, sigma means addition. Okay. 0.2 ln 0.2, like that you add. Ln values I have written here for each of the data set they have given. Substitute that, multiply, add everything and you will get 1.09 as the answer. Then next one, in a plant species the genotype capital D, capital D, capital E, capital E is crossed with all small letter D and E. And the F1 is test crossed. Okay. Considering that the two genes are linked and 20 map units apart in the chromosome, the percentage of the test cross progeny in the genotype small d small d small e small e is. Okay. Now they have told the two genes are linked and they are 20 map units apart. 20 map unit means the distance between these two genes is 20 map units, or I can say they can have the centimorgan, it will correspond to recombinance frequency. So 20% recombination will be shown. Okay, so the recombinant offsprings will be 20%. Parental will be, we have to calculate. Okay. So that is what is meant by 20 map units. So first what they have done, they have crossed this and this. So you get these are the gametes, and the F1 you get. The F1 you test crossed. So cross with the recessive parent. So F1 will produce 4 gametes. They will show recombination also. So how many gametes will show recombination? 20%. So 2 gametes will be produced. Okay, so each will be 10%. 20 divided by 2, 10%. So if 20% is recombinance, 80 will be parental. 2 parents are there, both will produce 40%, 40%. 80 divided by 2. So they have asked the percentage of the test cross progeny with the genotype DDEE will be. So that you will get from that DDEE is your parental combination, not the recombinant one. So DDEE will be 40%. See here. That is your parental combination. So answer will be 40. Then in a farm animal breeding program, the animal with the dominant A phenotype, the recessive B phenotype, dominant D genotype, recessive E phenotype are commercially important. So when you are breeding the animal, you will select those with capital A, small b, capital D, small e. Okay. Because they are commercially important. Then inheritance of these traits, they follow Mendelian laws. So independent assortment of genes is there. From the tetrahybrid cross of the genotype, see these are the genotype. All heterozygous for all the genes. The expected frequency of offspring that will show all the above mentioned desired phenotypes is. So that means the progeny having capital A, then a progeny having capital B, a small b, D and E will be selected. Now see here, what do you require? You need capital A, small b, capital D, small e. So capital A means it can have either capital A or capital A small a, because A is dominant gene. So it can have 2 genotypes. B is recessive, so only one type of genotype is possible. Whereas D again dominant, it can have capital D, small d or capital D, capital D. E is recessive, so only small e condition will be satisfied. Now you are asked to cross these 2 organisms. So what is the probability that you will get this combination? See here, capital A, and here it can be either capital A or small a, that is why I have given a dash. Then small b, small e, capital D and small d or capital D. Okay, now what is the probability that you will get? We will take one by one. Okay, first we will cross capital A small a with capital A small a from both the parents. What is the probability that you will get A phenotype? So when you cross these, you will get these are the offspring. So A is occurring in this and this. So 3 offsprings out of 4 will be showing the dominant A characteristic. So 3 by 4. What will be the for the phenotype small b, it is crossing capital B small b with capital B small b. So you will get 1 by 4 offspring. Similarly for D and E, you will get 3 by 4, 1 by 4. Now multiply all the progenies so that you will get this combination. So that will be 0.035. Then next one, a population of bacterial cell grows from 10,000 to 3 and 3, 6, 7, 8, 10 raised to 8 cells in 6 hours. The generation time of the bacterial population is. So you should know the formula NT is equal to N0 2 raised to N, where small n is the number of generation. Once you know the number of generation and the total time, you can calculate the generation time. So first you have to calculate number of generation. So number of generation you can get from NT equal N0 into 2 raised to N, or 2 raised to N is equal to NT by N0. NT is after time T the population size and N0 is the initial population size. So we know it is this is NT is 10 raised to 8 and this is 10 raised to 4. So you get 10 raised to 4, take log on both the sides, you get 40 by 3. I have just simplified this. I have removed the multiplied both sides with 10. So 40 and 3. Instead of taking 3010, I have taken only 3 for simplification. So 40 by 3. Then you calculate the generation time, that is total time by number of generation. So total time they have given 6 hours. So 6 hours you convert to minute. Your answer is in minute. So 60 minutes multiplied by 6 and divided by n, that is 40 by 3, you get approximately 27 minutes. Okay. Then in a sampling expedition near a peninsula, 180 dolphins from a large population of dolphins were captured and marked by tagging their dorsal fin. Okay, 180 dolphins they captured, marked by some color or by pricking or something, whatever way is available, or tying some something around the fin of the dolphin. The tagged dolphins were then allowed to join back into the population. Then they left the dolphins back into the water. In subsequent expedition, that is after 1 or 2 months, they again go for expedition and they caught 42 dolphins from the same area, and they were photographed from same large population, and out of these 42 they found that they have captured 7 dolphins which contained the tag. Okay. So assuming the population size remained the same and the tags were not lost, the estimated population size of dolphins in the peninsula. Now this is a mark recapture method, and there is a formula for calculating it. N is equal to MC by R. So N is equal to estimated total population size. M is marked in the first sample. How many samples you took and marked? That is M, that is 180. They have given here 180. C is the total caught in the second sample. How many samples did you catch in the second expedition? That is 42. Then recaptured tagged individual, and out of these 42, 7 were tagged. So they were captured again. So recaptured and tagged individual, that is 7. Substitute in the formula, you will get 1080. Then next one, the number of 7-letter words with or without meaning. Okay, it can be any letter, any words starting with the letter B that can be formed using the letters of the word biology is. So your first letter has to be B, but I, O, L, O, G, Y can be any combination, anything. See that O is repeating twice. Okay, now B is placed in the first position. The remaining letters, they can be arranged in the rest of the 6 places in any combinations. So N is equal to 6, R is equal to 2. That is, the O is repeating twice. So number of arrangements will be N by R. So factorial, so n 6 factorial by 2 factorial, you will get it as 360. Then next one, a buffer solution is composed of 0.1 molar acetic acid and sodium acetate. So here the conjugate base is sodium acetate and acid is acetic acid, which is a weak acid. So buffer is composed of acetic acid and its conjugate base. Now the change in pH of 1 liter buffer solution. So the buffer is total volume of this buffer solution is 1 liter. And you change the pH of the 1 liter buffer solution by adding 50 ml of 1 molar NaOH into it. So you have 1 liter buffer solution of acetic acid and sodium acetate. Into that you are adding 50 ml of 1 molar NaOH. Now NaOH is a strong base. It will neutralize this acetic acid in the buffer and form sodium acetate. So acetic acid concentration will decrease, sodium acetate concentration will increase. Okay, now what you have to find out? The change in pH. So what was the initial pH when acetic acid and sodium acetate were present, and what is the pH after addition of NaOH? They have given Ka of acetic acid. You have to calculate pKa first. Then use Henderson-Hasselbalch equation for further calculations. So first one, the pH is equal to pKa plus log sodium acetate by acetic acid. So pKa we have to find out. That is minus log of Ka. Ka value they have given and you will get it as 4.76. Substitute the pKa value. Log they have given for sodium acetate. Initially it is 0.15 molar and for acetic acid it is 0.1. You will get 4.936 as the initial pH value, that is before addition of NaOH. Now after adding NaOH, now moles of NaOH that you have added. So molarity is moles by volume. So moles is equal to molarity into volume. So they have added 1 molar NaOH. So 1 into 50 ml they have added. So 50 you will get 0.05 moles of NaOH was added. And what was the initial mole of acetic acid? We know the moles of acetic acid is 0.1 and it was 1 liter buffer solution. So will be 0.1 moles. Now NaOH is a strong base and it will react with the weak acid forming sodium acetate. Now final moles of acetic acid. I told you it will reduce. So initially it was 0.1 mole. Now it will minus 0.05 moles will be 0.05 mole, or that will be the final mole of acetic acid. But sodium acetate concentration will increase. So how much moles of sodium acetate was there initially? It was 0.15 mole. Then final moles will be add both these, this and this 0.05 moles. So 0.20 moles. Now what is the change in volume of the total solution? The first it was 1 liter buffer solution. Then you added 50 ml of sodium hydroxide. So convert it into liter. So it will be 0.05 liters. So 1.05 liter is the change in the volume. Now new concentration of acetic acid. You found out the moles of acetic acid, moles of sodium acetate. Now you have to calculate the concentration of acetic acid. Only then you can calculate pH value, because it is log concentration of sodium acetate by concentration of acetic acid. So moles by volume, you know the new moles of acetic acid is 0.05 by 1.05. That is the new volume. Similarly for sodium acetate also you will get this volume. Substitute the values, you will get 5.362. Now change in the pH. Initially it was 4.936 and now it has become 5.362. So minus both these, you will get change in pH as 0.43. Okay. Now a cDNA was synthesized from mRNA of a eukaryotic gene. Now cDNA synthesized from mRNA will not have introns, but cDNA will have UTR region, untranslated region, 5' 3' UTR. In between you will find the coding sequence also. So after cloning and sequence analysis, the double-stranded cDNA of 614 base pair. So total length of cDNA is 614 base pair. It revealed 125 base pair 5' UTR. See here 5' UTR 125 base pair. 120 base pair of 3' UTR. So here 3' UTR is 120 base pair. They have asked the number of amino acid present in the polypeptide encoded by this gene. So it is not having introns. So whatever region present between the UTR will be your coding sequence. So how much will that be? You add up 125 plus 120, subtract 614 from that answer, you will get it as 369 base pair. That is what is got here. So that is the length of the coding sequence. Now this coding sequence, it will have a stop codon also, which will not code for any amino acid. UAA, UAG, UGA will be there. Now there are 3 codons. So minus 3 you have to do. So you will get 366 base pair. Now you know that one amino acid it is coded by 3 base pair. It is codons that code the amino acid. Codons are triplets. So 3 base pairs will code for one amino acid. So 366 base pair will be coded by 366 divided by 3, that is 122 amino acids. Then next one. A rare genetic disorder resulting from homozygosity of recessive allele occur in 2 out of every 10,000 individuals in a population. So 2 out of every 10,000 individuals in the population is frequency of the homozygous recessive genotype, that is Q square, RR condition, small r, okay. So that is that Q square they have given. Assuming that the disorder is not lethal, nobody is dying because of the disorder does not impact reproductive success, no harm in reproduction, no new mutations are introduced, mutations are not being introduced, and the population is in Hardy-Weinberg equilibrium. So the percentage of carriers in the population that pass the R allele to the offspring. So you have to see for the carrier frequency. Now they have given Q squares, that is 2 by 10,000. From there you have to find Q. Q is the frequency of the recessive allele. So square root of that you will get this one. You know P plus Q is equal to 1. So P is equal to 1 minus Q. Now for heterozygous condition you have to see 2PQ value. So frequency of carriers, that is capital R and small r, they will be 2PQ. So substitute PQ and multiply by 2. You will get it as 0.0278. Now they have asked percentage of carriers in the population. So that will be 2.78%, or rounded to 1 decimal will be 2.8%. Okay. Then next one. The vector shown in figure, this vector has a promoter and RBS sequence. We already discussed promoter and RBS sequence is for expression vector in the 300 base pair region. So this is the 300 base pair region. It is having the promoter and the RBS. Between the restriction site for the enzyme X and Y. X and Y are the restriction sites. There are no other sites for X and Y in the vector. vector doesn't have any other x and y side. So if you digest vector with x, it will just open up. If you digest vector with y, again it will become open up, or I can say it will form single stranded. The promoter is directed towards y. So promoter will be somewhere over here near to x. It is directed towards y. Okay. The insert containing only an ORF provides. So this is the insert ORF, open reading frame. So this is the insert that you have to insert into this vector. So this insert, it is having 3 fragments. If you digest with x and y, that is 300, 200 and 700, and total it is 1.2 kb. This is the ATG or the starting site. So ATG is here, means promoter is here, so it has to be oriented in this way. So you will get x, y, x, x, y. That will be the orientation. Promoter is here, over here. So promoter has to synthesize this gene, means so ATG has to be coming near to the promoter. Okay, so it will be in x, y, x, x, y direction when you insert this gene of interest into the vector. Okay. The ORF is cloned in the correct orientation in the vector using the single restriction enzyme Y. So you cut this vector with Y and you insert this ORF into the vector. The size of the largest fragment of the recombinant plasmid expressing the ORF upon digestion with enzyme X is. So you have to see for the largest fragment you get from this recombinant plasmid, which is expressing the ORF once you digest the recombinant plasmid with enzyme X. Now when you insert this gene, this ORF into this vector, X will have 3 sites, 1, 2 and 3. Okay, so 3 digestions will take place if you digest with X. Okay, now see, I have drawn the whole thing over here. So this is the vector which was 3 kb earlier, and you inserted the 1.2 kb region here in this vector. So this is the one which is having ATG. This is your promoter RBS site over here, that is facing the Y side. So it will be in this orientation. So 300, 200, 700. So 1.2 plus 3 kb means your recombinant vector will be totally 4.2 kb. Okay, totally it will be 4.2 kb. Already it was 3 kb. When you inserted 1.2, it became 4.2 kb. Now you digest this with X. So X digestion will give you 3 fragments. That is 300 plus 300. Okay, this one. Till here one digestion. Then this region, that is 200, and this 700 XY, this region will not be digested. 700 will be part of your vector backbone. So your largest fragment is this one. Okay. The other 2 are smaller fragments. So totally you will get 3 fragments, 1, 2 and 3. They have asked the size of the largest fragment. Just subtract from 300 plus 300, 600 plus 200, 800. So subtract 4200 base pair, that is 4.2 kb minus 800, you will get 3400 base pair. So that will be the size of the largest fragment that you obtain after digestion with X. So that's it. Thank you for watching my channel. If you want still previous year question papers of IIT JAM Biotechnology, you can go to my channel Omni Bio Entrance. In the playlist you can see for IIT JAM, and you will find them there. Thank you.